Hello everyone, my name is Daniel Kent, and today is July 25th, 2020. This is my Case Connections 2 video. Today I've chosen to go over cases 2 and 3. We will start with case 2. In the information provided by the case question, there is a photo of an MRI and a PET scan of a brain tumor. According to the case question, the patient was given fluorescently labeled D-glucose. This caused the PET scan to show concentrations of glucose transporters, GLUT, as red shaded areas. When viewing the PET scan, it is clear that the GLUT is overexpressed in the area of the tumor. Glucose molecules are hydrophilic and require carrier proteins to cross cell membranes. GLUT proteins are the carrier proteins which facilitate and regulate glucose transport down a concentration gradient. The GLUT1 expression of the GLUT proteins are found in almost all tissues and are highly expressed by erythrocytes. GLUT1 proteins are the primary actors in the transport of glucose molecules across the blood-brain barrier and are thus very important when considering this case question. In other normal tissues, glucose transport is facilitated by other families of proteins. As seen in Table 19.2 of our textbook, in the liver, glucose transport is regulated by GLUT2. In the skeletal muscle and adipose tissue, glucose transport is regulated by insulin-dependent GLUT4. And in most areas of the brain, glucose transport is regulated by GLUT3. In order for cancer cells to survive, the cells must adapt. The most common way cancer cells adapt is through a mechanism called the Warburg effect. Dr. van der Heijden published an article in Science magazine detailing the effects of the Warburg effect. This effect describes the way cancer cells modify their cellular metabolisms to favor anaerobic glycolysis instead of oxidative phosphorylation, which is a preference in most other cells. When cancer cells utilize anaerobic glycolysis, energy is produced by the conversion of pyruvate to lactate through the means of fermentation. By utilizing the mechanism of the Warburg effect, the cancer cells achieve a rate of glycolysis which is far greater than the rate found in normal cells. However, it is also far less efficient. Anaerobic respiration provides only 2 ATP per glucose compared to the 36 ATP per glucose produced in the oxidative phosphorylation pathway. This results in an increased glucose uptake, which explains the increased expression of GLUT1 in the tumor area shown in the PET scan. This is actually beneficial to the tumor because despite being less efficient, anaerobic glycolysis occurs much faster than oxidative phosphorylation. That completes my analysis of case 2. Next, we're going to move on to case 3. In this case, a 5-year-old child presents with fatigue, polydipsia, and polyuria. When the blood serum insulin levels are drawn, the baseline blood sample of the child showed an insulin level of 0.8. where a normal insulin level should be 6 plus or minus 2. 1 hour after the glucose load was given to the child, the blood sample showed an insulin level of 5, where a normal range should have been 40 plus or minus 11. Taking all the child's symptoms into account, the child could possibly be suffering from anemia or diabetes. While the symptoms of fatigue and drowsiness might fit a diagnosis of anemia, it does not account for the polydipsia, polyuria, and the low insulin levels. When reviewed together, fatigue, polydipsia, polyuria, and the low insulin levels shows that it is clear that the child is likely suffering from insulin-dependent type 1 diabetes mellitus. In his current condition, the patient's fasting blood glucose levels would have a higher range than normal. The range would be higher because in type 1 diabetes, the cells do not uptake blood glucose due to a lack of insulin. This forces the glucose to stay in the bloodstream. Two regulatory enzymes which are likely active in this patient are glucose 6-phosphatase and fructose 1,6-biphosphatase. That completes my analysis of case 3. Thank you so much for watching my video. Until next time, thank you.